In 1961, it was hypothesized by Dr. Mary Lyon that since a homogametic individual has an extra copy of the sex-determining gene, one is not genetically expressed. This inactivation is random, and once decided, the progenitors of that cell will never express the genes on the inactivated chromosome. Instead, this defunct chromosome exists as a supercondensed structure called a bar body. This essentially eliminates all access by proteins that transcribe genes and has the effect of equalizing the amount of X-linked gene product in males and females. In humans, inactivation occurs in the late-stage blastocyst, shortly before implantation in the uterus. More precisely, this takes place in the group of cells called the epiblast, which eventually becomes the embryo. This random inactivation of a parental X chromosome in females has the odd effect of allowing some cells to express traits present on the maternal X chromosome and some cells to express traits on the paternal X chromosome. This phenomenon is called Mosaicism. Perhaps this is best observed in tortoiseshell cats. Since the gene associated with fur pigment in cats is located on the X chromosome, we can easily visualize the mosaic effect. Say, for example, a sperm carrying a paternal X chromosome marked XP that encodes for yellow fur fuses with an egg containing a maternal X chromosome marked XM, encoding for black fur. This cell will divide and divide and divide again, and at some point in each of these cells, one of the X chromosomes will become inactivated, leaving only one allele for fur color. This means that all of the pigment-producing daughter cells of these cells will only produce the black pigment encoded by the maternal X chromosome, while the other cells will only produce daughter cells capable of synthesizing yellow pigment. You can begin to see how this will affect the eventual phenotype of the cat. Interestingly, since this process is entirely random, even identical twins and clones would have different mosaic patterns. Now let us see if we can predict the phenotypic probabilities of the offspring of a yellow short-haired female cat with a black male who is heterozygous for long hair. First we will note the genotypes of these individuals, then we will complete two Punnett squares to determine the probable genotypes of the offspring. X big B indicates black hair, while X little b indicates yellow. Big H is the allele for long hair and is autosomal dominant to little h, the allele for short hair. Now we will cross X big BY from dad with X little b from mom. Half of these offspring will be yellow males, and half will be tortoiseshell females. Next, we will cross big H little h from dad with little h from mom. Half of these offspring will be heterozygous for long hair, and half will be homozygous for short hair. Now that we have our probabilities, we can construct our branch diagram. Of our yellow males, half will have short hair and half will have long hair. Likewise, any tortoiseshell females will have a 50% probability of long hair or short hair. We then multiply our probabilities to reach the likelihood of a given phenotype.